Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Bannattini. Here I interview brain scientists and discuss their work as well as the latest advancements and challenges in the field of brain mapping. Today, my guest is Dr. Bharat Biswal, who, while in graduate school, was the first to report the observation of functional correlation in the resting state fMRI signal. In this case, it was between the left and right motor cortex. This first resting state fMRI paper was published in Magnetic Resonance in Medicine in 1995 and is titled Functional Connectivity in the Motor Cortex of Resting Human Brain Using Echoplanar MRI. This observation back in the early 90s ushered in an entirely new subfield of fMRI and a new era in functional imaging that permeates much of what we do today. Dr. Biswal is a distinguished professor in the Department of Biomedical Engineering in the New Jersey Institute of Technology, as well as the Department of Radiology in New Jersey Medical School. He received his bachelor's degree from Utkal University in India in electrical engineering in 1989. He received his master's at Michigan Technical University in 1991, and he received his PhD from the Medical College of Wisconsin in the biophysics department under the guidance of Dr. Jim Hyde in 1996. So I've known Bharat since we were both graduate students back in the early 90s. And in this conversation, he recounts the events leading up to and including his discovery of the signal. We also talk about all things resting state fMRI related, including things like white matter correlations and potential clinical applications of resting state. He even asked me a few questions of his own. This is definitely worth a listen as he weighs in on the challenges, limits, and opportunities of resting state fMRI today. It was truly a captivating conversation and quite a bit of fun, as I always have with Bharat. So I hope you enjoy it. I have a special guest today, somebody I've known for years, Bharat Biswal, who I think all of you know the name, all of you know of his paper that he made that seminal discovery of resting state correlations in functional MRI signal. And he basically created an entire subfield of fMRI on his own. So Bharat, thanks for joining.
Thank you, Peter, for inviting. It feels going back 20, 25 years. In case anyone doesn't know, I started my PhD when the famous Peter Bannattini he was just starting his 4th year. And so I have been trying to follow up and still trying. So thank you, Peter, for inviting.
Now, your paper, I was actually looking at Google Scholar, your first paper has almost 10,000 citations. I think any of our first papers, even Ken Kwong's, I think is around 4,000. So yeah, it's gotten a lot of traction. And what's important as well is that, I mean, you've been really active continuously ever since, sort of pushing and refining and advancing resting state fMRI as well. So yeah, we both came from the same MCW institution, and it was a great place for fostering graduate students to just try stuff, and a lot of creativity. And of course, that credit goes to Jim Hyde, who encouraged us to kind of be bold in how we're exploring and not just sort of fall down well-trodden paths. So why don't we just start out by tracing the history of, like, for instance, you started at MCW. I'm just kind of curious why you chose MCW, and then also, as you were a graduate student, what caused you to go in this direction and make this discovery? So what was What was the environment like and what happened?
I had visited Milwaukee for a job interview. At that time, I had applied to a few PhD programs, and I was really interested to do a PhD. And I was staying with a friend, and he suggested that I meet Dr. Hyde, this famous physicist who was working on medical imaging. And maybe I would find that work interesting. So I went to visit. I called, at the time, I don't think email was that popular. So I called his office, and I think I called on a Friday, and Monday I went to his office. It was like in March, I believe. And so the PhD application was already completed. And Dr. Hyde still took the time to meet with me. It was Dr. Hyde and Andre. And Dr. Hyde, like right away said that, he typically picks only one PhD student, and he had already selected Beth Myron, who was coming from Yale, from John Gore's lab. And he was just talking, what I had done for my master's thesis. And then Dr. Hyde mentioned that when he was at MIT, there was an electrical engineer who was fairly good, and his name was Kalman. And I think I told Dr. Hyde, I think Kalman is beyond good. He's considered like his work is considered one of the foundation in signal processing. And so Dr. Hyde asked me to explain what this Kalman filtering did. So luckily I had taken some courses so I could explain. Then Dr. Hyde said, well, let me see, why don't you apply? I will see if I have the money or not. And then Dr. Hyde said, which other places have you applied? And I told him, and then Dr. Hyde said, I think you are going to get rejected from this university, and this university. You will only get admitted to this university, and maybe you will get a TA-ship at one or two places. And you may find it hard to believe, Peter, but maybe a week later, I started receiving the letter, just as Dr. Hyde had predicted. And so I also became a little curious as to, who would think that Kalman was just an okay guy. So I came back because I was still in Milwaukee. So I came back to MCW, read to the library, looked up some of his papers, and then read more and more about MRI. And I would also call him like every Tuesday that has a decision been made. And I think Chris Felix, who was the administrator, he one day saw me sitting in the library and reading MR textbooks. So I think he went and told Dr. Hyde, I'm just very fortunate that, soon after I received admissions to the PhD program.
And so once you were there, so I remember, I kind of faintly remember when you were starting, what did happen in terms of how you discovered, what were you searching for? And then how did you discover this? I mean, there's always a certain amount of serendipity. But,
right. So I forgot an important thing. So when I left that day after meeting Dr. Hyde gave me your paper. Right? Dr. Hyde said, well, here is this technique which has come being developed, and Peter is doing, and this is the first paper. And maybe you may have done signal processing in one dimension, you may have done some images in 2 dimension, but here is a problem in 3 dimension. 3D plus time. And so the way he said was people are using fMRI, but using the same method as PET. And if fMRI has better spatial and temporal resolution, there should be something unique there. So this could be a good thesis topic. So, I think this was almost like his exact words, this is what PET has done. This is the resolution of fMRI. Look at this data and see what do you see. And so what I did was, at that time, I again, I didn't really know much about neuroscience. So the only thing I knew was about EEG. And so I read up some literature. And when I was reading, I thought, and there were some papers where they were saying that when you performed a task, the respiration and heart rate would change just around that time. So I thought that would be like a simple problem to do, that Peter has done finger tapping, and for some other task, maybe if I looked carefully when the task was occurring, then I would see the respiration change or maybe get delayed. And so that is what I started, but it wasn't that easy because first of all, we didn't really have that many data points to look at, like each epoch of respiration cycle. I could just get maybe a couple of data points and then investigate. I thought the other thing I could try is to maybe look at the brain as a transfer function. You have an input, you have an output, and then can I better characterize the transfer function? And if I could characterize the transfer function, maybe we could model it better. Again, in signal processing, the best input that you can send to a signal is a random noise. And so I was doing that, but I also thought it would be good to have a resting scan, just as the null distribution. So I had like a resting scan finger tapping paradigm as you had done. And then I was asking the subjects to do finger tapping in a random fashion. And then again, collecting a rest scan. But the transfer function wasn't really working that well, because for longer duration, I could predict quite well, but for shorter duration, it didn't work. And then I was looking at the spectral features, like what noise were there and so on. While the heart rate was aliased, I could see the respiration, but it looked like the respiration had some power, but not a whole lot of power. So even after filtering the respiration, there was not much of an improvement. I thought maybe I might as well see what the structure of this noise is. So I had I correlated from the sensory motor cortex as you had done. And then I did the same thing for the resting state scan. And I should say that I was a little surprised, but not very surprised, because I thought, if you have an electrical circuit or if you have a system, then if you present a stimulus and you get a response, then if you present a noise, then you should get a similar response.
Yeah.
During that time, I would meet Dr. Hyde once a week. But before I met Dr. Hyde, I would speak with Zeren Yatkin to make sure that the results I was showing him was all fine and things like that. So after I showed her, so I had made some points, like the noise inside the brain is greater than noise outside, which is to be expected. Like there was difference in noise power between gray matter, white matter, and CSF. And then there was this correlation that I was observing. So Zeren Yatkin said I should stop everything else and just focus on this one. And so I give her a lot of credit, because she said she was a clinician. So she said maybe that this could be something like what people do in EKG or so, that you could just collect the data at rest and then you would have this pattern that could be used.
Yeah, that's cool. That's so Zeren was prescient in that regard. So, and pretty much all you were doing then was just trying to characterize the filter properties of the time series by looking at the spatial and temporal correlation and whatever. Yeah, and you just found instead sure, that the other motor cortex was more highly correlated. And so Zarin jumped on that. So I'm kind of curious then, when it sort of grew, so you showed hide, and then it grew from there.
Right. So I showed Dr. Hyde in maybe like in my first year, like February of my first year, these results. And then like Dr. Hyde wanted to make sure that this was like very like everything was done correctly and so on. So like one thing we were worried was, I think I forget whether it was you or maybe Steve Rao, had done a study where you were imagining finger tapping and showing some response, right? So we were worried whether there was some kind of effect from that. So we tried to use different parameters. So we would tell the subject that, okay, you're going to do an auditory task where you would be listening to some words or some music, while we would tell them that we would be collecting the resting state scan, and then such that there was no imagination of finger tapping.
Yeah.
And after we had collected the resting state scan, we would go inside the scanner and say, hey, can you do a finger tapping? And most people, like, because many of them were graduate students who had already volunteered for your study, for them it was fairly straightforward. They would say, oh, no problem. So we would do a finger tapping task at the end. So that was one change we had to do. The other change was, whether it has some noise, whether there was respiration or cardiac aliasing. We were worried about that. So that is why we decided to only do 2 slices, but with a much faster sampling rate, and then do that. After the first observation, to the time I had a manuscript written, took maybe another 6 months or so. Okay. And in the meantime, we had gone to, I think, ISMRM in New York in 1993. There I had shown that there was this noise characteristic difference between gray matter, white matter, and CSF. At that time, like I should say, at that time, you were doing finger tapping, Alan Song was doing diffusion. So I was still not sure what my thesis topic would be and whether it would pan out or not. And so I go to the conference, and the first talk was by Jack Bellevue. And I think the second talk was by Robert Weisskopf, who was showing these differences between gray matter and white matter. Yep. And at that time, I was in my first year, so I had no idea who Robert Weisskopf was. And I think I asked you or Eric, and the response was like, he's one of the top 5 MR physicists, right? So, so yeah. After that, I guess I thought, okay, maybe if Dr. Weisskopf is doing, maybe it is okay to go along the same lines. Initially, the paper got rejected, I should say, from a couple of journals. And then, I was like getting desperate to have a topic and get it published. At that time, your paper had also come in MRM, so we submitted to MRM. But unlike your paper, my paper went through 4 rounds of review.
Oh God!
But I'm glad it was published.
Yeah, no, and what's interesting is, I think that a lot of people were. So you sort of simultaneously also were working on just understanding the noise characteristics, and people hadn't also fully appreciated, physiologic noise, and so you sort of helped push that part of the field too. But what I found really interesting is that even after you published your paper, I'm actually, it's amazing that few people picked up on it, but it really took it seems like it took about 10 years for the field to sort of like finally catch up. And if you follow, like, you ever do a search on, like, resting state or whatever, you have a few papers after 1995, it's sort of trickling, and then it explodes at like 2005 or 6. And then, obviously now, in the past few years, OHBM has been dominated by resting state. But why do you think it took 10 years, about a decade of like not really quite catching on?
Right. So that's a little difficult question to answer, because like when we started, it's not like there was no controversies in the field. Unfortunately, in my case, it's not that the people disagreed with my data. It is that
Right.
people did not believe my data. And, I think that was like where the problem, like my problem started, was that people did not believe the data. And then initially when I would go to conferences, I remember after one talk, someone came and asked me how they could replicate my results. And I was just like, I was like a second or third year grad student. And I had like this famous person is asking me how they can replicate my result. And all I could say is that it's in the method section of the abstract. But the second thing was this, then after this person, another person comes and asks the same question. I didn't know what to answer. So there was a lot of that, but I feel that maybe it was like people maybe had used task activation. And therefore, if you truly believe task activation, then you have to believe, in my opinion, that you have to believe that the baseline condition is just random noise. And if the resting state is providing a functionally connected region, then what does task activation mean? Perhaps for those reasons, people were like very dismissive. As regards to why it took 10 years, I think things happened in progression. So after 1995 paper, Mark Lowe published.
Yep. Yep.
And then during that time, I had published a couple of other papers showing how you could use hypercapnia to modulate these fluctuations. I had another paper where we had used spin echo, simultaneous spin echo, gradient echo to show the relative weighting between flow and bold effect.
Yeah.
And then there was a paper I had on Tourette's syndrome, which was again like Zen and Yetkin had, I was involved with her on that project. So I had like these work. And then I think what happened was Michelle Hampson published a paper in Journal of Neuroscience.
Okay.
I think that was the first like high profile paper using resting state. And then there was the famous paper by Marcus Reichel and Deborah Gusnard, I believe, where they had used the default mode they had described. And then in 2003, there was a paper by Vinod Menon and Mike Grecius in PNAS, where they said, okay, there is this default mode, but you can basically use the same analysis as Biswal et al. And get the same pattern. And then came Mike Fox's paper in 2005. And I think around that time also, there were many papers on default mode, which was getting published in PNAS. I think that certainly helped. And then I believe in 2010, the paper with Mike Milham and others, I feel here that it was not just a paper. We had made all the data public. We had also made all the codes public, right? So in other words, what we were saying is that, okay, this is all the data we have. This is all the scripts you used, and you can run this script to generate this figure. So, if there is some error in the method, find it, or else, you can use it for your resting state data. I think that was maybe, all this, I would say, all the papers I mentioned, and I'm sure I forgot a few other important papers during that time, but all of them contributed.
Yeah, no, actually, also, and that's a sort of a good segue. I mean, that paper was kind of ahead of its time, I think, a little bit in terms of open access, open data sharing and sharing code. That's the paper Toward Discovery Science of Human Brain Function and PNAS. There were like 1,000 connectome runs or data sets and so what has become of that paper or that data set? Is it still being used, or is it, I'm sure probably people are downloading it still.
I believe it's still one of the most downloadable data sets from the Netric website. There are many software, right, which is maybe a few megabytes, but this is a huge data set which people still download and use. Mike has, of course, done a whole lot by using the monkey data set. Data set. There is, of course, now HCP data, both with healthy subjects and so on. In terms of that paper, I should say that we were like very, very lucky, but also we got a lot of help from many people who I can never thank enough. So when we initially thought of the idea, we had simply written email to maybe 50 people, anyone who had published a resting state abstract or a paper. I wrote to them. And I think at that time, it was even a crazier idea than doing resting state, is to asking people to give their data. And so many people said no right away. There were few people, I should say, few friends who had to call and beg that please share the data. I just need some data. And that is where later I found out that Yufeng Zhang, who was at Beijing, was trying to do a larger project. So when we asked him, he gave us 200 data sets. So wow. With 200 data set, we had more than 500 data. We thought that we could have a paper. And at that time, we also had asked Randy Buckner, without realizing that he was trying to do this, the super structure project that he did. And so he was kind enough to, give us a similar number of subjects. So we had like 700 subjects. So then we went back to all the people we had asked and asked them that, hey, we already have 700 data. So we really want you to participate. Can you share the data? And so we got another 300 data sets, believe it or not. Wow. So that is how it was done. And I think like everyone who gave 20 data set or 30, or Randy Buckner and Yufeng who gave 200, I will always be grateful to them. And I should say people had also tried before trying to make data public. So maybe like I was very lucky to be at that time. And I'm just like also quite happy that nowadays most journals, they make you have this code and data sharing section. Yeah, and people are much more open.
Yeah, no, actually, and it helps the field. It pushes the field. It pushes the, people can download the data, try their analysis, advanced methods, and actually, and it's still being pushed to sort of, advance standardization and acquisition as well, and all kinds of things. But so, all right, well, let me switch gears a little bit and talk about the resting state signal in general. Certainly it's been advanced a lot over the years. I mean, there's dynamic resting state, there's clinical studies. You've done a lot of work on trying to look at clinical populations, how they differ, trying to tease out more information from the resting state signal. You've also worked on, it seems like kind of the thing that you think about a lot is filtering methods, various types of filters, spline, homomorphic, adaptive, diffusion. So what do you think is this, as far as the state of resting state fMRI right now, maybe if you could just highlight the big advances or some of the major advances, and then also sort of talk about a little bit of what your contribution or some of your more recent work along those lines. I mean, I actually, it's funny, I was looking at some of your papers and I saw really cool stuff. Like, I saw effects of weather on resting state, effects of, and then you actually have a paper that shows connectivity in white matter. And I would love to talk about that as well. But just in general, just looking at psychiatric disorders too, it seems like an interesting thing. So wherever you want to take it.
Can I first talk about white matter? And then we had looked at the gray matter. And for a number of reasons, like people would dismiss the white matter as not like there should not be much signal. And so then there was this famous paper by Laura Lewis, where she has shown that in CSF, in sleep, how you can have this modulation. Yep. And I had like a very similar data set with me, and I hadn't even looked. So I was thinking that, what are the assumptions we are making? And have I, while I had like compared to task activation, while I had assumed that maybe in resting state there is information, could it be that I should be looking at white matter and other specific areas? And when I started doing some literature search, I found out that there were a couple of papers already. So I cannot say I was the first person to investigate white matter resting state. So there's some very nice work by Michael Peer. There's another work by Ziguao Ding from Vanderbilt. So in our case, I simply focused on the corpus callosum, because the corpus callosum is the largest white matter region. And basically using the same diffusion atlas as has been used, and using those parcels to look at connectivity. And what we find, which is again very surprising, is that there is a good concordance between what we get from white matter tracks and what we get from using resting state from the white matter. And maybe, again, it still remains to be seen. I'm trying to see whether the white matter signal and the gray matter signal could be combined, and then we could find regions like parcellations, which could basically map out the whole brain and not just the gray matter regions. That is like what I'm trying, but there's still a lot of work to be done.
Yeah, I mean, certainly, people argue that white matter doesn't have enough blood, or the hemodynamic coupling is different, but or maybe the signatures of the resting state are just different. And so it is interesting. Your maps and your paper looked convincing. So if you take like a seed voxel in white matter, other areas along the track show up, but not gray matter though. And that's kind of interesting.
Well, actually, the way we analyze now, I have focused only within white matter, because I wanted to first establish, are these white matter signal organized in some specific way? So now we know how the gray matter is organized. The next try is to see if they can all be combined together to find, and is there some kind of interaction between the white matter signal and the gray matter signal, or are they from the same origin?
Well, that's interesting. I sort of think of white matter tracks as maybe, at least from my naive view of the brain, it's like, I imagine they're super highways and they're always active in some way, but I'm sure they're modulatable. And that sort of leads to sort of a question that I had, what your perspective of, you had one paper sort of maybe talking a little bit about the nature of, like, is resting state signal, is it like oscillatory, or is it driven by, some people argue it's driven by like these punctate quasi-random activations that are just co-activations. What's your perspective now on like what's driving the resting state? Is it purely oscillatory? You can do Fourier analysis and see it, or is it punctate activation? And if so, what part is what role does it play ultimately?
Peter, that's a very difficult question because if I was to ask people doing EEG, how are the EEG patterns generated? Why are they oscillatory? And what is their content? Even after 100 years, people may not have, a very good idea. So in terms of what is the nature of the signal, I still think that these low frequency fluctuations maintain some sort of balance between blood flow and metabolism. And that is why we have these fluctuations. And that is why when people have tried to use anesthesia or even hypercapnic stimulus, these fluctuations get disrupted. Unfortunately, it's not that easy to change the blood flow without changing metabolism and vice versa. So it's not that easy in terms of, whether it's completely oscillatory or is it quasi-periodic. That is, in my opinion, a little difficult because one of the researchers whose work I admire most is Sheila Clyhouse.
Yeah.
And according to her, it's quasi-periodic. So I would say it is periodic, but I would also say that it has these period, while it may have a dominant frequency, but it changes around that same frequency, perhaps the same way as our heart rate, right? We may have a heart rate, but over the course of a day, it may go up and down by 10%. So similarly, maybe it does.
So what about, so in one of your papers also, you kind of suggest that there are different classes like low frequency and less low frequency or higher frequency. Right. Have you seen patterns related to that?
Right. So I think the paper, I think Xinyan Zhuo was the first author. But in that paper, Peter, what we are doing is we are looking at specific frequency band, and those bands were you derived using EEG literature. So there's a paper by Kuribuzaki where he basically made a power spectrum of the human EEG signal and showed that there is slow 4, slow 3, and alpha, beta, gamma, and which are present. So with the multiband and other sequence that is currently available, that was what we were trying to do, show that whether, what are the different frequency component. Also, is there any other, beyond 0.1 hertz, which we have typically filtered out. If we use frequency beyond that, can we see the same network pattern? While we do see network pattern, it seems to be maybe much more diminished, maybe like 10 to 15% of the power is in that high frequency.
And sort of along the lines of understanding resting state, what are your thoughts as to, well, one, what information do you think what information is it, like, a combination of sort of cognitive sort of conscious thoughts and then, subconscious processes, or like you were saying, purely physiologic sort of oscillatory sort of neurons just need more oxygen, or is there other, I mean, I'm sort of, maybe I'm thinking of what the purpose and what information is there to tap into, to use from resting state. So sort of thinking of, what there's been some papers recent one by Maurizio Corbetta's group talking about sort of these unconscious sort of priming processes that resting state satisfies.
Right. So if I understand your question correctly, you're asking like what function does these functional connectivity serve? And I would say that perhaps the fact that there are distinct resting state networks which have been associated with different function, perhaps that shows that these networks are indeed related to different cognition. And I think like Lucina and others using like the salience network and others have shown how that can occur and how that can be compromised, that could be compromised in clinical populations like schizophrenia. So yeah, I mean, I do think that, the resting state network does affect cognition and our cognitive processes.
So you've been spending a lot of time on, I mean, not really a lot of time, but there's several papers that you have on ways of filtering, of methods of cleaning up the signal. Is there what's the best method right now? And then the other part of the question is, do you think that, I'm always hoping that this is the case, that we can fully clean up the time series, get rid of all the physiologic fluctuations that are not related to, something meaningful about neuronal processes like, respiration, alias cardiac. Do you think that there's hope for just cleaning up the signal completely with the right choice of adaptive filtering, perhaps?
Maybe we can hope, but it may not be that easy, simply because how we sample the noise will determine what kind of noise and how noisy our signal will be, right? The kind of filtering I had used in, let's say, in 1990s, I would not use those these days, because now we can collect so many data points. It's, again, I should say, I would not use, I would say there can be improvement, simply because at that time we could maybe hope to collect 200. If we had 200 data points, that was very good. Now with the Human Connectome project, we have 1,200 data point, maybe more. And with multiband or even the newer method like MREEG that Jürgen Henning and others are developing, you can collect 10 data points per second. So for those kind of cases, one can use different filters. So perhaps based on the data or based on the MR data that we collect, we could design what kind of filter we want. Also, as Andre would say, what is the signal we are interested, right? If we are interested in task data, then the filter would be somewhat different than if we are interested in the resting data. So I think based on that one, design.
Yeah, no, it's something I'm always kind of pushing my own lab on trying to figure out ways of more cleanly separating the neuronal connectivity from just physiologic stuff going on. And it's a challenge. You're right. It depends on the acquisition. It depends on the TR. It depends on a lot of other factors that we still, and it also varies over the brain too.
Right. So can I ask you a quick question? So
Sure.
I should just say that, in this podcast, just to continue our conversation like we were having in grad school, although Peter had suggested he would send me a question, I told him no, because it would have made me much more nervous. And so we can just have a discussion. The question I had is, where do you see is going to be the limits of spatial and temporal resolution in fMRI, and how much data can be collected?
Okay, you're asking me that. So, okay.
Yeah.
I was going to ask you something like that. I mean, with the risk of sounding short-sighted, I actually think that, at 7 Tesla, I've seen results now that show 0.4 millimeter resolution. I think the limit in resolution obviously is with time, but also in the phase instability with multi-shot stuff. If we can figure out better ways of doing phase correction to account for physiologic noise, we can go down to lower than 0.4 millimeters because we'll have time and we can fix that, all the phase errors with multi-shot. So I think the images can go down to below 0.4 millimeters. And I do believe that, the signal to noise obviously is almost zero. I mean, not zero, but it's like about 10 to 1 at 20 to 1 at the most at 0.4 millimeters, even at 7 Tesla. We could filter things out, but we're already in the thermal noise regime. So it's, there's techniques such as Nordic that are coming out that might clean up some of the thermal noise, but a little bit less than 0.4 millimeters. And then functional resolution, if we can be specific to capillaries, which I believe we can, we'll be able to with the right sequence, it'll be about 0.4 millimeters, enough to resolve most of the layers in fMRI, and also whole brain. So that's my kind of long-winded thought, but.
Right. Thank you.
But okay, so I was going to ask you as well, not really necessarily or did you want to ask me any more questions? I'm fine.
No, it's fine.
Yeah, but no, that's good. This is fun. This is fun to go back and forth. I was wondering though about networks and about, you've also written papers on dynamic connectivity, looking at changes in connectivity in states. And I always thought that, the brain, how finely can you divide up states of the brain in time, and how finely in space are functional areas divided into networks? I mean, so we have a paper with Renzo Huber where we go to, super high resolution, we look at resting state, and we can pull out digits in resting state. It seems like that's kind of a limit in some way, but maybe not. But also in time, as you were saying, this, this power law relationship, things are potentially changing pretty rapidly. If the more finely you look, the more rapidly they change. So you have these states that are like 5 minutes or 2 minutes, and then they go down. What are your thoughts on that? On temporal and spatial delineation of resting states? How far do you think it goes? How far do you think that we can look?
Right. So again, Peter, you're asking me like really tough questions. So which is fine, because I don't know the answer, but it makes me perhaps think a little bit. In terms of brain states, there is work by Elena Allen, where she's taking series of images and then showing that there are like 7 brain states. Even in neuroimaging, brain state means different things to different people. To me, brain state is, or I should say state, is state space is a term which is used in system identification. So if you have a dynamic system, you find the number of states it has, and they are useful because you can predict what will happen using a combination of these states. And the way Elena Allen has done that is using the same identification toolbox or same identification methods. The way like Katie Chang and colleagues they have done is looking at the dynamics and how it changes from time point to time point. As to what is the optimal resolution is a very difficult question to answer, because it is almost asking what is the optimal window length to be used in a filter. If you use a broad window length, then you lose the resolution. If you use too short a window length, then you will be increasing noise. I think, again, I have, Vince Calhoun has written a few papers. Well, of course, no one can really read all of Vince Calhoun's paper, but in terms of window length, there's a couple of papers where I believe maybe 40 seconds, I could be wrong, was the amount he had mentioned.
Yeah.
So perhaps around that. But again, I would say based on the resolution, because, in Neuroimage not too long ago, I read this really nice review from Switzerland about micro state EEG.
Yeah.
I'm forgetting his name. I'm so sorry. So
yeah, that's right. That's right.
Right. So there they had used the EEG data to look at micro states. And again, in that they can show, or they're showing that they can tell schizophrenia from healthy control based on this. So I think for answering this, the temporal resolution is critical to answer. Right.
Yeah.
I hope I answered your
question. No, that's that. I mean, I think that, sorry about the questions. I mean, it's sort of like purposely trying to find things that we're all struggling with as far as that's concerned. And I don't know that, I don't know the answer to that. It's funny, when you were asking me questions, I thought you were going to ask something embarrassing or something, because we have lots of stories as far as graduate school, but which you're welcome to as well.
Someday I'll write a tell-all book.
Oh no! Oh no! Neither of us have anything to hide anyway. So,
right.
I'd be remiss if I didn't talk about clinical applications. I mean, you have some kind of nice review articles and other things sort of going over potential clinical applications people find, and along with your groups, your group and collaborators as well, have found differences in networks and resting state networks with ADHD, autism, bipolar disorder, schizophrenia, depression, OCD, substance abuse, Alzheimer's. And this is sort of the thing I'm confronted with more and more, is that people find these differences in these groups. And then usually, the general public ask me about this. They say, well, why isn't this used clinically then to help diagnose and understand? I'm like, well, because, when you put a person in the scanner, it's just one person, and it's hard to make, it's hard to find a robust biomarker that has a large enough effect size for an individual. And resting state is great because you don't have to have the subject do anything. Maybe looking at movies might be better a little bit. You have a paper sort of on that, which I tend to agree with as well, but there might be just different though. So what are your thoughts on the ultimate, can there be a day-to-day clinical application of resting state, either we're doing nothing or looking at movies or something like that? What are your thoughts on that and what we'd have to overcome?
For clinical application, there's a lot of work which is going on, but as you said, can we do single subject determination, and using a single subject, can we say whether this is Alzheimer's or this is schizophrenia? I feel that slowly we are coming to that point, in a sense that once we describe that, okay, if in Alzheimer's or in aging, these are the networks which are going to be different, then you can use, and you had a large data set of subjects, then one could use like a machine learning algorithm to find the difference. You could also use partial least square, like methods that Randy McIntosh and others have developed, where if you know what the group level changes are, then you can look at individual and see how much they deviate, and then perhaps make a prediction.
Yeah.
But for those, I feel you need like not 100, but maybe data sets in like 1,000 or so. And then again, age and other factors come into play. But I think it should be possible. But having said that, clinical diseases are so heterogeneous. They are always typically more heterogeneous than healthy control. Just like 2 days ago or so, Tom Insel had written this editorial in New York Times expressing maybe some of his frustration and like hope what can happen in psychiatric disorders. So to me, if psychiatrists are struggling themselves to classify the different types or subtypes of, let's say, schizophrenia, then resting state, perhaps I would be obviously very happy, but perhaps just resting state may not be that useful. One should use resting state in conjunction with clinical measure to come with a solution.
Yeah, no, you brought up a really good point. That the idea of the, and it's sort of similar in cognitive science too, the ontologies of, or how we divide up clinical disorders. And it's sort of, agreed upon, but the brain may not manifest in the same way as the disorders, as the behavior is looked at. And so, you might have an ontology of different disorders, but then the brain might have its own pattern of how it is disordered that might not, be in line with those. So yeah, that's a struggle. That's a struggle. I was actually just listening to Russ Poldrack talking about in the 1800s, we talked about things like, suavity or things like that, which doesn't really have a neural component. And maybe even these disorders may not have, they might not be as cleanly defined as manifestations in the brain, but that's tricky. But yeah, there's always hope. And I'm glad you brought up the partial least squares with Randy McIntosh. That I think that there's hope in doing that type of approach if we get a big enough effect size.
Yeah, I think there is also like some work where they have like some other group have done PCA to also do. So I think like one can go along that route also. So
just to move towards, looks like we're starting to get towards the end, but this is great. I could talk for hours. We'll have to have you back. Or maybe we could just have a podcast devoted to our stories from graduate school. What are some of the most exciting things you're working on now? And what do you see as the ultimate future of resting state fMRI? I mean, these are sort of open-ended questions, but what excites you most about resting state right now that you're working on, and what are your prospects for the future?
I think I mentioned a little bit about what I'm doing, like white matter, also PPI analysis, looking at the interaction between different networks. Predicting the future is very difficult. So who knows, right, how things will pan out with resting state. If I was to guess, I would say that, like machine learning is becoming very popular. There's so many very nice studies coming up where they are showing how machine learning is useful to find subtypes or characterize resting state. The other area that I'm really have been trying to follow is trying to look at like the neurovascular coupling and how it may modulate resting state. So during grad school, I had done work with Tony Hudetz on these work. And now I think Elizabeth Hillman, like her work is really good. Even Arvind just recently had a paper in Nature Methods, again, which provides unprecedented spatial and temporal resolution using multimodal information to better understand these neurovascular mechanism or origin, if you will, of these fluctuations. Also, it's like, can we look at different parameters within resting state? For example, ALFF seems to correlate with glucose consumption. ReHo correlates with local connectivity. So if we use many...
So just to clarify, ALFF is the measure of total power of bold signal in the low frequency range, and ReHo is
the regional homogeneity.
Yes.
So ALFF, thank you, I should have, yeah. So
that's right.
ALFF is, it basically tells us how much relative power is in the low frequency band, while regional homogeneity tells us how each voxel correlates with its neighbor. So again, using a number of different measures of resting state, could we gather more information than simply doing, let's say, ICA or seed-based analysis? So
yeah, and actually you bring up a really good point. I mean, there's, so many people are, I mean, I think the tendency is to think of just a certain ways. Oh, you do ICA, you split it up, and or you can do, PCA or other sort of time frequency. But there are other measures, like you mentioned it, ALFF and REHO, that really pull out aspects of resting state that could be more informative about disease. Yeah.
Right, and I think, as I have told, we had a few papers in which we had shown how, if you cannot do breath holding, which you had first done, hypercapnic stimulus, then you can substitute the resting state. It's not the same, but it gives you, at least in healthy subject, a measure which correlates. So, I always say, even if you don't believe in resting state, you should collect the resting data, because if nothing else, you can use it to calibrate your task activation data, and maybe share it with others who are interested in resting state.
That's good advice as far as that's concerned. Any advice you'd want to give, or is there anything else you want to say about resting state or about what you're doing? And then lastly, what would be any advice would you like to give to, young scientists, maybe the next person that will discover the next big thing?
With young scientists, I would say that the field is so much more open now. Anyone, with a good, decent computer and modem can do fMRI for it, and for young students, anyone who is interested in fMRI, first they should buy a famous book by Peter Bandettini, right? Read it in the first few days. And then, there are many, like 26 controversies that they can think carefully. There is 10 questions about resting state. They can, ask those questions. And, if they need to, I hope they would want to learn more. There is another fellow graduate student, Alan Song. He has also written another book with Scott Hutel. It may take them a little more than reading your book, Peter. So
mine was light.
Yes, but it gets them started, right? And so again, I'm not joking. I'm serious, because one can really get started, and I think initially, people may find it hard to believe when some journals published MRM or NeuroImage, when the issues came out. I would open and look at the color pictures, how nice they looked, right? But now it's hard to imagine pictures which are not in color. And even there are so many more resources. One could just use YouTube and find good lectures and so on. So I feel there's a lot of problem that still is open for people to solve. And I think also, again, I think it doesn't apply to you, Peter, but perhaps more to me, where, now the new student there, language skills and other things are so much better than when I was at their age. So it's a really exciting time for students.
Yeah, and I just have to maybe re-emphasize for everyone is that there's a lot of I think that there's a lot of serendipity involved. I mean, for instance, you were looking, when you made your first discovery though, you were kind of looking for something else in some sense. You're just trying to understand the noise characteristics. You're doing that, but then you saw something unique and you noted that it was unique. I think that a lot of things are potentially interesting that are right in front of us. And it's just a matter of, doing what you did, is sort of like stepping back and saying, hey, wait a second, this is interesting. So I think that in itself, I think could be something to learn from you as far as that's concerned.
Thank you, Peter. So can I ask one more question?
Sure. Uh-oh.
So,
I'm always nervous. When you say you're going to ask questions, I get nervous.
No,
Yeah.
that's not true. But so what do you think is the role of multimodal imaging? Where do you see it can help, either task or rest?
The first answer could be, simultaneous multimodal imaging, can capture a temporal domain or a spatial domain that's different than whatever modality you have. Like, fMRI and EEG is the first thing that comes to mind. EEG can capture more of the temporal domain. And there's some evidence potentially that, feed forward versus feedback signals are different frequencies in EEG. And so that might help constrain or enhance the interpretation of fMRI in that regard. And ultimately, what we really want to do is not just descriptive. We want to build models. And to build these models, we have to actually have as much detailed, fine-grained information to feed into it as possible, along with whole brain information. So the fMRI gets great whole brain information. All these modalities might add their components, either in the temporal domain or in the, whether it looks more directly at neural activity or not, that might complement. So it's sort of a synergistic sort of combination of like, you can more deeply interpret fMRI if you have some other technique in sort of supporting it. But also, I think that the information you get can be a little bit more than each technique in itself. There's a big role for not only like EEG and fMRI, but even not simultaneous, like MEG and fMRI, or even optical imaging, to answer some of these questions about, calcium imaging, like with Elizabeth Hillman. There's some really good insights from that, the idea that it's more excitatory related than inhibitory, potentially. So things like that, I think are, and also of course, optogenetics,
Right.
a huge amount of control. So, so there's things like that. You can manipulate the experiment, or with TMS, you can modulate nodes and look at how it changes resting state. So there's an added dimension of what you can do and how deeply you can interpret things with multimodal. But yeah, that's at least my off top of the head description of what we can gain from multimodal. But anyway, is there anything else that you wanted to talk about? Anything else that comes to mind?
If I can just ask one more thing, then, so do you think when, like, it's something about movie watching, we are like, again, we are very interested, but I always have the question that, is movie watching the same as resting state? Let's say two people watch the same movie at the same time. Will their experience be the same? And if not, then is it the intrinsic network which is creating the difference, or is it something else? What is your opinion about like movie watching in general?
Yeah, I look at like movie watching as equivalent to like in cardiac, doing cardiac studies where you give a stress test. You sort of have to see differences. You kind of have to push the system a little bit. And so I look at, certainly you can see differences with resting state. Movie watching is tricky because if it drives the system like too much, then people will be like in lockstep and they'll all be similar potentially. But there is a chance that you could drive differences. Like with Emily Finn, when she was here, she did, and even before she came, she did like a, listening to a story, and some people who were slightly paranoid interpreted the story differently. And then that was drawn out where it may not be drawn out intrinsically with resting state. So that's the hope, to build these movies like they're litmus tests or stress tests to pull out these differences that may not show up. But it's not perfect. And I do think that what you look at, what you experience consciously is related to the movie watching, but there might be real information in the resting state that's not time locked with the movie. That's also a biomarker. That's also potentially interesting. So that's part you're still missing. So yeah, I think it helps. I don't think it's like clearly better or clearly worse. It's just sort of, it helps. It's complementary. I would always recommend you're sort of doing a task though, potentially along with doing the resting state. I guess that's my best explanation of, it's like a stress test. It'll pull things out, but it won't pull out everything. So.
Yeah, there's a lot of interest now in movie watching. So, and we have also doing some work, but since you have published a few papers recently, I wanted to get your opinion.
Yeah, I'm hopeful. And I think that, Emily is just cranking as far as that's concerned. She's really developing this whole science of what is the best stimulus that pulls people apart, that pulls different, that maximizes this effect size of differences between individuals. And there's, if you really get into it, there's a whole science to what you can do to someone to make them appear different than others, or to be, to pull out the differences. So, which is a really cool thing that she's doing as far as that's concerned. So, and we're certainly working on it as well, but in our group still, it'd be fun to combine and movie watching. Oh, I forgot to mention, this is probably one of the most important things, is that it opens up the type of analysis you can do. You can do sort of cross-subject correlations that help pull out these networks that may not have a perfect hemodynamic response, or might have perfect they show up as cross-subject correlations that may be correlated or not, depending on whether the subject's similar or not. So, and I'd love to apply that to layer resolution as well.
That is exciting area. I wish I could get some data on layer fMRI.
Well, all of the data that we collect, we try to make publicly available. And Renzo Huber has a whole data set on resting state, or actually watching the Connecto movie with the same time locking. He has like 25 data sets already on the Open Neuro.
Okay, I'll check it out.
Yeah.
I thought I had seen there were only 2, but I haven't checked.
Yeah, I think there's more. I think he had to redo some of them because there was an artifact in some data sets, and he had to redo it, but it's there. He's working on it. But anyway, so thank you again, Bharat, for coming on and having, this has been a great conversation. It's always really fun, not only it just reminds me of old times and catching up, but I always like to, I think your thought process is creative and unique. And I think that's, it's always fun to hear what your perspective is on these things. So thanks again.
Thank you, Peter. It's a real pleasure for inviting. Most of the discussion I used to have with Peter was typically after midnight, after, either of us had finished scanning, and then we would chat. So also, I would like to, if it's possible, take this opportunity to, thank one doesn't get much opportunity in public to thank, your mentors like Dr. Jim and Karen Hyde, Ted Dio, Tony Huditz, Zaran Atkin, Bob Cox. I'm sure I'm forgetting some name. Sijang Lee. Also, I would say I was in Wisconsin, I was even fortunate to have, many illustrious alumni like Peter Bandettini, Alan Song, Eric Wong, Beth, Jack, and many others, which I learned as much. So Peter, thank you again for inviting.
Neurosalience is brought to you by the Organisation for Human Brain Mapping. This week's episode was produced by Ekaterina Dobryakova and Alfie Wearn.